Hi, and welcome back to biology. Today we're going to talk about the cell and the corresponding concepts and theories about it. So, cell is considered as the basic unit of life, di ba? So, muna balik-balik na to nga na definition of the cell because it is the smallest unit that can carry out the processes of life. Remember in our biological organization, it is actually the cell that is capable of carrying out the processes of life. Kadto nga to ang fundamental properties of life. Below the cell, the organelles themselves, the molecules, they don't possess the life kasi kulang ang kadtong mga properties to consider them as living thing. And above the cell, those are more complex ng mga levels where life may occur. So I hope we have some understanding about it. Now, organisms are actually made up of either one or more cells, di ba? So unicellular or multicellular organisms. So for example, we have the bacteria, the archaeans for the unicellular and some protozoans. And then we also have, the humans, we also have plants. We are higher complex organisms which are made up of many cells, trillions of cells. So what are those basic functions that are carried out by the cells? We have first is the material transport. So yung passage of substances between cells and within the cell, those are a kind of material transport. Yung osmosis, diffusion, things like those are part of this concept. We also have obtaining and using of energy. So, obtaining, of course, yung sa pagpasok nung mga substances, we have to have that source of energy. Like, for example, humans, we have the glucose. And then it is broken down. And while it is broken down, you have the byproduct, which are the adenosine triphosphates, which are then broken down through hydrolysis na naman para there is a release of energy. Okay, so balik-balik na siya. We also have waste disposal. So when you say waste disposal, it has something to do with the worn out organelles which are digested by hydrolytic enzymes, which are inside our what organelle? The lysosome. So yung lysosome na yon, one example of hydrolytic na organelle, they have to clean, di ba? So para walay mga anomalies. Like for example, mga problems with the genetic makeup of an organism, they have to clean that one. They have to hydrolyze the nucleotides para dili na masagol during replication. So we have also replication, which is intended for reproduction. So we have to replicate our DNA, and then of course response to their environment. Anyway, so the cell is first coined by Robert Hooke, a British scientist. So we will talk about them, including other scientists, about their contribution during our assessment. So you will be searching other scientists for that, including Leeuwenhoek. So, yun. And I think the most prominent icon, kumbaga, sa biology or sa science is the microscope. So, microscope has revolutionized the way we see life, di ba? And from a very primitive microscope like here, we were able to have these things here. So this is Jessa, my student from 2019. She is studying insects. So she is using a dissecting microscope, and this one is another light microscope, which is one of the basic kumbaga na type of microscopes nowadays. This is from FSUU, so we will be discussing later on the parts of microscopes and how we are going to use it. Kasi, di ba, we are having this pandemic, and sad to say, I think we will not be able to use them. And I hope nga bisan virtual, you can still understand the mechanism of microscopes. So microscopes are one of the very important things in biology, especially when studying cells. So there are large cells, like for example, the onions, the root tips of an onion are being used for basic studies in cytology. Kasi dagko iyahang cells, so makita nato dito, especially during mitosis, yung mga cells, they have the stages of cell division. Okay? So now, what are the postulates or what are the proposals of the cell theory, kumbaga? What are the principles? First is, all organisms are made up of cells, either unicellular or multicellular. So we will talk about them later on with the scientists that proposed these things, okay? So of course, we also have that the cell Tao is the basic unit of life. We all know about it. And cells come from pre-existing cells. So sa una, they thought that cell spontaneously appear out of nowhere, okay? But they were able to see that no, cell doesn't come from emptiness, kumbaga. It has to appear from something else, which are of course those cells that are already existing, and they exist through cell division. But of course, if you will going to trace back in time, like unsa na tong mga epoch before, cells came from non-living things, but they don't appear out of nowhere. Okay? Dili pasabot nga mura na siya ni appear bulagaan na dayon. It doesn't work like that. It took millions of years para na-cell. Okay? And it has to be part of an evolutionary history of organisms. Kasi cells make all living things nga, di ba? And cell is the basic unit of life. So dili siya basta-basta mugawas lang out of, emptiness. So wala lang tapos nigawas na siya. And in fact, napatoy mga proposals before that humans are born like a miniature nga version and then we just become adult over time. Dili siya ingana. Okay? Mga lang pagtuo before. But we will be dealing with these concepts in the next lesson na to. But I'm just giving you an overview para dili ta. Well, I know that some of you are familiar with these concepts, but that would be a good thing kung mabalik na to. So, okay. So we have these organelles. Sorry for that technical problem. Organelles are known as little organs kasi during the researches of the pioneering scientists, 1800s, 1900s, they were able to see that there are things inside the cell, and they term them little organs. Okay, so they can be seen through a very expensive, of course, and high-tech na gid nga microscope, which are the electron microscopes. So we will talk also about electron microscopes later on. So unahon na to si light microscope and then si electron microscope. Of course, here in FSU, we don't have an electron microscope kasi it's very high-tech. Nana siya sa mga hardcore laboratories. So basically, the electron microscope uses electrons, which are beamed through a specimen, and you could see the very up to nanometers na siya nga unit of measurements, like very nipis na gid kaayo nga na mga objects. So sa cell organelle, we try to deal first with the cell membrane. So this is only present in eukaryotic cells. Okay? So eukaryotic, we will also talk about eukaryotic cells later on. So eukaryotic cells, we are eukaryotic organisms kasi our cells are membrane bound. Unsa man ang membrane bound, meaning naputos siya sa membrane. And what is that membrane? We call it a cell membrane. And even other organelles are also membrane bound, like the nucleus, the endoplasmic reticulum, Golgi apparatus. Those are coated with a membrane. Okay, and this membrane is actually made up of two layers of phospholipid, okay, oriented to opposite sides. So if you're familiar with phospholipid, na siya ay phosphate nga head and lipid nga tail. And sa ato ang phospholipid bilayer, those phospholipids are oriented to opposite direction. So head, tapos between them is the tails. Okay? So because of that, naatay ginatawag na selective permeability of that membrane. Kasi it has something to do with the chemistry nga ay kini kay like charges, they can come in, kini kay they don't attract each other, they cannot come in, and they have to have another way of coming in through the cell. Things like those are because of the chemistry of the membrane. Remember nga si phosphate is polar while si lipid is non-polar part of that molecule. So here, in this diagram, you could see that here is the phospholipid bilayer of the membrane, the plasma or the cell membrane. Same lang na ha? Plasma or cell membrane are the same. Okay? And we also have here, if you could see, like a very simple diagram of the phospholipid bilayer. We have the polar head, the phosphate head, which is hydrophilic because it attracts water, of course. And then we have the fatty acids, the lipid, which are what? Hydrophobic nga part sa ato ang membrane. And dito pod, dire sa obos bali, naapod siya tail pod of a different napod nga phospholipid, and then oriented on the other side is its hydrophobic head. Okay, and it is also embedded with proteins and carbohydrates, which we collectively term as, some are glycoprotein. Glyco means carbohydrate. So, ingana na siya because daghan po charges, different charges ang mga proteins nato. So, naalay certain molecules that can come inside the channels kadto mga protein channels within the bilayer, the phospholipid bilayer, the membrane. So, naa dito, those are channels where the molecules can come in and can come out. Okay? That's the cell membrane. We also have the cytoplasm. So, si cytoplasm is said to be a jelly-like substance made up of water, salts, and proteins, which we collectively term as cytosol. Okay? And all other organelles are suspended in the cytoplasm. So makita nato diri that this just imagine that the hollow spaces, okay, the hollow spaces in this diagram is made up of cytoplasm. The cytosol ang substance, specific substance niya ato. Okay, so ingana siya ang cytoplasm, and everything is suspended daw in the cytoplasm. Okay? And here is the nucleus. So, si nucleus is only present in eukaryotic cells. Wala siya sa prokaryotic cells, and you will know why in the next few lessons. So, it is enclosed by another double membrane na pod, just like sa ato ang other organelles. It is enclosed by a membrane. And that membrane is actually, kung familiar mo sa endoplasmic reticulum, actually, kana siya, is directly connected sa ato ang endoplasmic reticulum. Okay? So, it houses the chromosomes, yung ating genetic material. So, yung mga pores, if you could see the pore, those are actually not technically pore lang a pore. They are actually tunnels where the products of nucleus would go through the tunnel and dito siya sa endoplasmic reticulum mo. Adto. Okay? So here, the endoplasmic reticulum, so it is said that it helps move substances within the cells, and it is made up of interconnected membranes daw. Okay? It has two types, rough and smooth ER. So, interconnected membranes, which is directly connected, of course, from the nucleus. So first, we have the rough ER. So, naay mga ribosomes that are attached to in the rough ER, sa surface niya sa gawas, Here, sa sulod, we call them as lumen. Okay, lumen. So if you could see, this is the nucleus. This is just, shall we say, a diagram, a cross-section diagram. Pero if you're going to, to imagine it as a whole, if this is the nucleus, ang ER nakabalot sa iya. And those bangag-bangag sa nucleus are actually direct na siyang connection sa ER. Okay? And katong mga products of transcription nato nga mga mRNA would go through the ER. Musulod sila diri sa lumen sa ER. Diri kani, musulod sila diri and then they would be gathered to the ribosomes didto sa ato ang ER nga surface to be processed to have a protein nga product. So that is how important the ER is. So membranes are produced for the cell and its expanding membranes form transport vesicles. So imagine na ninyo, when you say transport vesicle, this is also similar with the Golgi apparatus. Okay, the rough ER and the smooth ER. So how do you imagine this one? Think of a bubble and then the product is katong hangin nga imong gihuyop. Okay? Imagine nga surface ni sa ER and then you blow the air, and that air, hypothetically, a product of transcription, or no, a protein. So, mubad off na siya dito sa surface sa ER, enclose dayon tong something, katong product, enclose siya sa bubble, that is the transport vesicle. And then, muadto na po na siya dito sa Golgi apparatus to be processed. Mo fuse na po tong bubble sa surface sa Golgi, and it will be processed by the Golgi to either become another organelle or a certain hormone nga mugawas sa plasma membrane. Pag mugawas sa plasma membrane, katong kinatungyan na pod sa Golgi, mao na pod to ang mubad off na pod, tapos mo fuse na pod to siya nga surface sa vesicle didto sa plasma membrane, and then mugawas na pod siya didto, and it will be excreted by the cell. So actually, those are mga connections sa ato ang mga organelles. Okay? So we have the smooth endoplasmic reticulum. So these are the function of the smooth ER, carbohydrate, tsaka lipid synthesis, and then we also have mga detoxifying nga mga property. Anyway, we won't be dealing much on the smooth kasi mas tagaan na to importance si rough tsaka the Golgi. So the Golgi, so ito yun, this is what I'm saying. So for example, diri atong product sa ER, katong protein coming from the ER, inside the vesicle that came also from the ER, it would fuse in the surface of the Golgi. So naatay terms ana sa surface of the Golgi gihapon, and then after it fused inside the Golgi sa iyahang lumen, it would be modified, and once it is finished, it would come out inside another vesicle na pod. So ingana siya, murag mo pop na pod siya, and that vesicle, kung hydrolytic enzyme na siya, that would be considered now as a lysosome or any other organelle, or if it is made up of mga watery substances, it could be the vacuole na. So yung ano na siya, and if it is something na kailangan by other organs, it would leave the plasma membrane, the cell membrane, para madala siya dito. So unsaon man na siya, so katong pinch or bud of nga vesicle that contains that molecule or substance would fuse to the surface of the plasma membrane na pod, and mo leave na dayon to siya nga molecule. So here, if you could see sa illustration, you could get nga the product of the nucleus, katong transcription nato sa DNA, it becomes, or it would transcribe an mRNA, and it would leave through the nucleus to the endoplasmic reticulum. Okay? So I hope na sundan. And then that product, mahimo na siyang ma-convert na siya or ma-translate into protein, and that protein could be something like structural or something like globular transport protein, or any other proteins. Could be something na pang-digest, enzyme na siya nga pang-digest o mga other molecules, things like those. And then motong ingay na nga kana siya nga product would leave the endoplasmic reticulum through a vesicle. So mo cleave na siya, mo cleave ang surface sa lumen and sa ER, sorry, and then that would leave to the Golgi for any other modification. And magsunod-sunod na siya sa cisternae or sa surfaces, sa stack sa Golgi, and it pinches off to the other side. And if it's something na needed, like this one, it's although it's not something na naka-disclose on siya nga molecule. So basically mura siya enzyme, mura siya kanang mga ginasecrete nato nga mga molecules sa mga glands. So mo na lang tong molecule nga magawa sa ato ang cell. Okay? So kani siya on the fifth step, katong vesicle mo fuse na pud na siya dito sa plasma membrane, and the molecule would leave or excreted na dayon sa cell. So just keep in mind nga everything inside the cell are membrane bound, and ganong needed ang membrane para na ay connection, indirect or direct na connection ang mga products of each of the organelles. And that's how important the cell membrane is, at least in eukaryotic organisms. So we will discuss, kung wala pa kaayo na klaro for some of the students, we will discuss this one in our synchronous session, okay? So here the vacuole. Vacuole is just a derivative of the Golgi din eh. It's something like another vesicle lang gyapon ni siya. Okay? And ang sulod niya are water, nutrients. So kadto siya coming from the ER. Sa smooth ER usually, those things mo-process na siya padulong sa Golgi, and ang di na siya magawa sa plasma membrane, instead magstay na siya within the cell for the storage of those molecules. But it is just another vesicle gihapon. Another one is a lysosome. Lysosome is just a derivative gihapon sa vesicle sa Golgi. So coming from the DNA nga nagbuhat atong enzyme katong genetic information of the cell, it transcribed na siya into an mRNA and into a protein. That protein is specifically an enzyme, a digestive enzyme, meaning hydrolytic or tingbungkag na siya other molecules. That's how important lysosomes are para sa clean up sa mga worn out ng mga organelles or other substances. So dayon, that product of the ER would go into the Golgi, and after the Golgi, muagi dito the modification, mahuman na siya dito sa cis nga phase sa Golgi, and then sa mahitabo, that becomes our lysosome na, and that lysosome will engulf those things nga dili needed sa cell. And in fact, when you say digest, it is just to say nga it is broken down either chemically or mechanically. But in this context, usually chemical na siya nga digestion of an individual or an entity, either waste or mga invaders, mga bacteria for example. Sa ato ang mga macrophage, sa atong white blood cell, daghan tag lysosome within those cells, kasi needed ang lysosome to do the job of the macrophage, or in general the white blood cells. Kay daghan may klase sa white blood cells pod, but almost the same lang sila og mga function. And then we also have the mitochondrion, or plural mitochondria. So it has its own DNA. Kasi naay history pod ang mitochondria with respect sa eukaryotic organisms. Okay, naay history ani, and you will know na it is a bacteria-like organism that was engulfed by another cell. Okay? And instead of digesting the mitochondrion, gi-incorporate niya ang function sa mitochondrion to take it as an advantage to thrive on the environment. That's why na siya own DNA kasi it is supposedly not there. But because of some instances before, according to researchers, they were able to see that the mitochondrion is far more primitive than the eukaryotic organisms themselves. Okay? So it is also coated by a membrane. So the products of mitochondria basically are still enclosed by a membrane. So katong mga glucose na to, mga pyruvate that comes inside the mitochondria for ATP synthesis must be within the vesicle gap. Dili sila basta-basta mosulod. They are still coated or enclosed by a vesicle. So, it is important in cellular respiration. So, cellular respiration is basically just an opposite of photosynthesis. So, sa plants and other organisms, photosynthesis ang tigbuhat og energy nila. Sa ato, we also have the cellular respiration. Something like that. Okay, so the first thing is the glucose or other sugars, but basically it is prominently glucose. Glucose is still a sugar, huh? And then we also have fatty acids. So, in the absence of glucose or any other sugar, it opts to use energy coming from the fats, coming from the fatty acids. But basically, it is still the same molecule at the end of the day. Katong mga acetyl-CoA, mga pyruvate, those things are still being used to make ATP. Okay? But there are different pathways, manggod. We call it pathway. Kung asa mo agi, like, it is more efficient to make ATP using glucose kasi. Kaya mas dali man bungkagon ang glucose over other molecules. And it releases ATP, of course. So that ATP, na siya potential energy, that when it is broken down, mabungkag na siya into an ADP and a phosphate ion, what happens next is na ay ma-release ng heat, and that heat fuels everything inside the cell. Mao nang mulihok ang mga organelle, mga molecules within the cell. And it makes us alive. Mao nang, over time, once the person is dying, magkabugnaw ang lawas kasi the ATPs are getting low in terms of their level, in terms of their count. Wala na kaayoy mabungkag nga ATP. And that's it. Of course, the chloroplast. So, it is present in other organisms, prominently plants and other algae. So, it has a pigment called chlorophyll, which functions to what? To trap the light, di ba? The solar energy. And it produces glucose. Okay? So, ang glucose, daghan po nag-pathway dayon, it could become a starch or become a cellulose, ana. Kasi monomer man na ang glucose, di ba? Simple sugar. So, magka-stack, depende sa configuration, it becomes a cellulose. And then dito po sa fruit, for example, it becomes a starch. Sa banana. And then, over time, mag-ripen na siya, mahimo na po siyang sugar. That's it. So, thylakoid are basically just kaning mga sacs here inside the chloroplast. So, yung ana lang na siya. And we will deal more about them later on. And of course, the cytoskeleton. So, it is composed of microfilaments, microtubules, and intermediate filaments. So, asa man diri ang microfilaments here, and then the microtubules are here. Basically, these are proteins, okay? Cytoskeleton is just made up of proteins which are being produced by the ribosomes gihapon. So, dili na nato i-complicate ang things. So, that's why I'm saying before, kadtong sa atong biomolecules, that all things inside the cell are actually just biomolecules, macromolecules, proteins, lipids, fats, and nucleic acids. Nga, because of their different structures, configurations, arrangements, they have their own property. But basically, kung imong himay-himayon ang ilahang mga structures, they are just made up of those things. So, actin and tubulin, those are names of proteins. So, kani sila, these are strong enough to resist breakage, but soft enough to resist stretching. Ing ana siya ka resilient when it comes to its structure. So they are needed to provide support sa mga organelles that are suspended in the cytoplasm. So that's it. That's how cytoskeleton work. Okay, so during the development, during sa cell division pod, they have their own mga responses. Like for example, once nag-divide na ang nucleus within the cell, ang mahitabo sa microtubules is that katong mga spindle fibers, they would pull the chromosomes away from each other, the katong mga sister chromatids, para ma-turn into half ang number of chromosomes and it becomes haploid for each of the daughter cells. That's how important the cytoskeletons. Spindle fibers kana sa ato ang lower biology are just derivatives of the cytoskeleton pod. Okay? Which are made up of proteins like the tubulin and the actin.